Hey everybody, what's going on? You ready for Bioengineering 101? I'm pumped. Are you pumped? Let the cloning begin, right? We're all ready to have the cloning get started and happen? All right. So, if you've taken the Bioengineering 101 class, um, you should have done all the experiments to get up to the point where we start genetic engineering, culturing bacteria, growing bacteria with plasmids, and all this stuff. So now in week 5, we're finally going to get into genetic engineering and do some experiments. An experiment called a transformation, where we put a piece of DNA inside a bacteria, right? Now, it's kind of weird that we use bacteria, I know, and people might wonder why we use bacteria, why scientists use bacteria. Generally, bacteria are used in genetic engineering because they grow fast and they're easy to work with. Uh, they're easy to put DNA inside of, so you can see results pretty fast. For instance, if you're working with an animal, it can take months. Plants, it can take months or years. Um, these things take a long period of time. So, generally, scientists work with bacteria because they grow fast, they replicate fast, they're easy to use and work with DNA. So, scientists use bacteria a ton. It's like the workhorse of genetic engineering because you can replicate DNA inside them, right? If you grow up a big bunch of bacteria and they all have a piece of DNA that you gave them, and you purify that DNA from the bacteria, that's a lot of DNA you can get, right? So scientists use them all the time. Now, one of the things that we're going to discuss today, and is, it pretty much the main way to do genetic engineering, is by using plasmids. Plasmids. We're going to talk about that today. Actually, what I might do is first do a little bit of some drawing, so then I could just, kind of explain what's going on with the plasmids and DNA and genetic engineering. Then, if you haven't already, make sure that you download SnapGene. SnapGene is what we're going to use later today to look at what's actually inside of plasmids and how plasmids work and all that good stuff. So while you're following along today, go download SnapGene. SnapGene Viewer. It should be free. All right. All right. So let's say we got a bacteria and it's got a flagella and it's just like swimming along and stuff like that. So the bacteria has its own genomic DNA in there, right? It's really big. The genomic DNA of bacteria, it's like 3 million base pairs, let's say. Now, in science, there's this thing called a plasmid. Now, plasmids naturally occur in black bacteria. A plasmid is a piece of DNA that's generally around 2,000 base pairs. To maybe like, they can be pretty huge, but generally they're between 2,000 base pairs and like 20,000 base pairs, right? And the genome of the bacteria is like 3 million. So, so what happens is these bacteria will store these plasmids because they could transfer them easily between different bacterias. They can pick them up. It's just something that evolution has created that allow bacteria to transfer genes, you know, between species and between other bacteria really easy. So generally, when you're working with plasmid DNA, the goal is to get the plasmid DNA inside the organism, whether it's bacteria or yeast or human cells or fungi or animals, whatever, right? You're trying to get the piece of DNA inside bacteria. So that's all you're worrying about. That's your only goal. Once the piece of DNA is inside bacteria, it has genes on it that tell the bacteria to replicate the plasmid and do certain instructions that you want it to do. And people code that themselves. So they code the DNA, and you could code the DNA yourself um, to get the bacteria you want to do. Now, generally, the ways to get the plasmid inside bacteria involve adding certain chemicals. Um, the chemicals that are usually involved are polyethylene glycol in bacteria and yeast. What PEG does, polyethylene glycol, is it helps open up the bacterial membranes, right? Because polyethylene glycol is a polymer that will interact with the non with the lipid membrane of the bacteria. It kind of creates pores and holes in it. And this helps the DNA get inside the cell, right? Usually there's also another chemical which is a positively charged cation, and bacteria use calcium chloride, and yeast use lithium chloride. And what this does is plasmid DNA, any DNA, is usually negatively charged, right? And so the positively charged cations, right, calcium, lithium, what they do is they help neutralize the charge of DNA so it becomes more inert, and that makes it easier for it to travel through membranes and things like that, right? So generally, those are the two big parts of the transformation, PEG and calcium chloride. All you generally do in most transformations, whether it's human cells or bacterial cells or yeast cells, is mix the plasmid, the organism, and some chemicals. And that's literally it. And then you just let them do their thing. Um, and that's what is involved in a transformation. All right. So the transformations that you're going to do are very similar. Um, what you're going to do is you're going to mix some bacteria. The fresher the bacteria, the better. The fresher the organism, right? The organism's healthy. Um, it's going to be robust. It's going to keep growing. So fresh bacteria, and mix it with the bacterial transformation mix, and mix it with some plasmid DNA, and hopefully at the end you get a GMO, right? So what is, first off, do we have any questions? Any questions on any of that stuff? That makes sense? You just got some DNA and you're just trying to shove it inside an organism. Shove that DNA inside the organism and get the organism to use it. Basically, when you're doing transformations, what you're trying to do is, kind of like hack the organism. You're trying to convince the organism that the DNA you're giving it is its own DNA, so then it'll replicate it, it'll make the proteins, and it will think it's its own DNA. That's basically the goal. And we figured out kind of like standardized things that you need to include for this to happen. But that's basically the goal of the transformation, is tricking the organism into thinking that it's own DNA. So when you're trying to use DNA between like bacteria and yeast and humans, generally it won't work, right? So bacteria have different control elements in their DNA than yeast, than plants, than animals, right? So all these organisms have different control elements in their DNA that allow the DNA to work properly, to tell it exactly what to do. So a piece of DNA, as we learned about, isn't just like telling you that it should make a protein. There's so much more that actually goes into it, and we'll talk about that in a second. Yeah. All right. So let's look at SnapGene. So download SnapGene. I think on the Google Classroom there should be some DNA sequences. Download the DNA sequences, open them up, and let's look at what a piece of plasmid DNA looks like. I have this set up properly. So this is a piece of plasmid DNA. And this is what we call a plasmid map. Right? So a plasmid map contains all the major DNA elements that are on the plasmid and which direction they are being transcribed. So which strand is being used. So if it's going this way, it's the top strand, right? If it's going this way, it's the bottom strand. So it's easy to figure out what these things are. We can just look at the sequence, right? And see what's actually coding for it. So we can tell here the methionine amino acid is coded for by the codon ATG. It's on the bottom strand going in the 3' to 5' direction, right? So that's that gene. And now if we go over to this one, actually that's an origin of replication. It's not a gene. If we go over to this gene, you can see the same thing, ATG, except it's on the top strand and it's going in the opposite direction, right? So, but you don't have to worry about that stuff too much. That's probably more technical stuff that you won't really have to need to know if you're just getting started in genetic engineering or doing genetic engineering with plasmids or things like that. Generally, you just want to know where your gene is, and that's it. All this other stuff in the plasmid is not that important. Well, it is, but it's not. So a plasmid is composed of a few main things. Generally, what we call everything that's not the gene that you put in, so in this case, we'll say the gene we put in is the green fluorescent protein, we call it the plasmid backbone, right? And the plasmid backbone is just all the junk that you kind of need for the plasmid to work, right? It's just all that extra junk that's sitting around that makes the plasmid work. And so it generally you're not going to care too much. The one thing that you will care about is the antibiotic resistance gene. But the antibiotic resistance gene is tells you how you select for your genetically engineered bacteria. So let's talk about this a bit. All right, let's go back to this diagram or drawing program. It's paint. I know it's paint. Don't hate, don't hate. I like paint. All right. So how does a genetic engineering experiment work? Now this is the case for most all organisms, right? So generally you have like a whole tube filled with bacteria, right? These are all little bacterias inside our tube. We got our little microcentrifuge tube, and it's filled with bacteria, right? Now we add our plasmid DNA, right? And that goes in there. So the plasmid is obviously not going to make it into every single bacteria. There's a certain efficiency of the transformation. So only some of the bacteria are going to be engineered. Well, how do we know which bacteria or yeast or human cells get engineered? The thing is you include an antibiotic resistance gene. And usually people write this as like ampicillin resistant, kanamycin resistant, or something like that. So what you do is after you add the DNA and mix it with the bacteria, you put it in media, generally a Petri plate that has some antibiotic in it, right? Ampicillin. Now what happens is if you put the bacteria on here, the ones that didn't get the DNA are going to die because they don't have an ampicillin resistance gene from the plasmid. The ones that got the DNA are going to live because they also have the ampicillin resistance gene from the plasmid. So it's a really simple assay where you can determine, what is genetically engineered and what is not genetically engineered. Now this same technique is used for everything. It's used for plants, it's used for human cells, it's used for yeast, it's used for everything. You use antibiotic resistance genes. It's just the way that is easiest to kill off all the cells that aren't genetically engineered. It's as simple as that, right? So antibiotic resistance genes are in almost every single plasmid that exists. All right? Do we have any questions? No questions so far. Okay. Let's go back to the snap gene. All right. So we got our DNA sequence right here. We got our plasmid map. So really the major thing that you are going to be interested in the backbone that you're going to care about is the antibiotic resistance gene. What is the antibiotic resistance gene? So you can use that specific type of media to select for bacteria that have been, or an organism that have been engineered with this plasmid. So besides that, all you care about is putting your gene in this plasmid, right? Antibiotic resistance. Sorry about, I'm a little, stuffed up. My allergies are kind of acting up today. Sorry about that. So yeah, all right. So we can go through here and figure out everything that's going on, all right? So we got our antibiotic resistance gene. This one is for ampicillin. You can also use carbenicillin. This ORI is the origin of replication. What it does is it tells bacteria to replicate this plasmid. Yeast plasmids can also have yeast origins of replication. Generally, every plasmid, it doesn't matter the organism, is going to have a bacterial origin of replication because you replicate the plasmid in bacteria, right? You can grow up large amounts of bacteria, and when you grow up these large amounts of bacteria, you can purify the plasmid DNA from them to use in other organisms. Otherwise, you can imagine trying to, like, say, synthesize a piece of plasmid DNA is going to be expensive, you know? So we have learned to just replicate plasmids in bacteria. You can replicate them in yeast or something like that, but generally you're not going to get as much. Bacteria have the ability to create very high copy number inside their cells. So that means each bacteria doesn't have just one plasmid. Each bacteria might have hundreds of plasmids inside. Okay? So there's so many different copies of the plasmid inside the cell. Now, the origin of replication, depending on what origin of replication you use, there's different origin of replications. The bacteria will create a different copy number of the plasmid in the cell. So there are what are called high copy number plasmids and low copy number plasmids and medium copy number plasmids. Now, what these copy numbers are, generally, it really depends what you're trying to do, right? If you're just trying to make, say, like a single protein and as much of it as you possibly can, and the protein is not harmful to the bacteria at all, generally you want a high copy number plasmid, as high copy number as you can get, so that each bacterial cell will make as much of the protein as possible because there's so many copies of that DNA laying around. Now, it's not always the case that you want a high copy number plasmid. Sometimes you want a lower copy number plasmid because what you're trying to do is you're trying to express some protein that may be toxic. You're trying to express multiple proteins that are part of some metabolic pathway, and like what you mainly want is you want the protein that is the rate limiting step to be expressed really high, and the other proteins to be expressed lower because they don't matter so much. So there are cases where you may want different copy number of plasmids, but generally you're probably not going to deal with it too much. I don't think in my whole career so far I've ever tried to use a low copy number plasmid or something like that, like on purpose. So, you may or may not, but let's just say probably not, will not have to worry about that. So, what's after that? Now, generally, plasmids will have what's called a promoter. A promoter is a region that tells the RNA polymerase that it should bind, and it should start making the mRNA for this gene, right? So a promoter promotes the production of mRNA for the cell. There are different types of promoters that create more and less. There are also things called enhancers and other things that can contribute to the production of your gene of interest, your protein. But, again, generally you're just going to try to find a promoter that makes a lot of protein. It's generally in biotechnology, it's pretty one-dimensional. You're just trying to make a lot of something so you can sell it or use it or something. Now, after that is a ribosome binding site. Ribosome binding site tells the ribosome where to bind the mRNA, right? If you remember from last class, we talked about how the ribosome makes the proteins. So the ribosome needs a binding site so it can make the protein that will become, will make the protein that is your gene of interest. In this case, it's green fluorescent protein. Now, I want to say every protein starts with a methionine and ATG, but it's not completely true. There are, promiscuous stop codons that aren't methionine, and there have been a lot of data that shows that not all genes are started at methionine. But generally, when you are making a plasmid, when you're trying to express a protein or a gene that you have, you're going to start each protein with the methionine. You're going to use the start codon ATG. You're going to make the methionine, and that will start the amino acid linkage together to form your protein. Now, when you want to end your gene, you're going to have a stop codon. Here you can see it by a little star. Generally, that indicates the stop codon. So again, generally, you're going to use a stop codon. I think it's 3. Oh gosh, don't quote me on this. I haven't looked at the codons lately. But there are a few different stop codons. TGA is one of them. Obviously, it's being used in this gene, and it tells the ribosome where to stop adding amino acids so that the protein is going to stop being made, right? But it doesn't tell something like the RNA polymerase where to stop. So generally, you're also going to have a terminator. So you're going to have a promoter and a terminator. The terminator is going to tell where transcription, where the mRNA should stop being made, right? So like you have 2 different processes. You have the mRNA being made and you have the protein being made. Both processes need to tell when to start and when to stop, right? So you could kind of think of this as like a somewhat of a programming language. It's not 100% exact like a normal computer programming language because we're dealing with biological things that have promiscuity based on thermodynamics. So it's not going to be 100% exact, but it's going to have a high percentage of working this way. So both have the beginnings and the ends, and that's the important part to know. Now, generally, when you insert a gene into plasmid, all that stuff is already in there. The only thing that you're adding is your gene. So you're going to insert your gene like something between this NDE1 site and this BLP1 site or something. Now, what are these NDE1 and this BLP stuff? All right. Let's talk about that. Now, instead, molecular biology is complicated, and we're just starting to get to the place where we can synthesize DNA at a cheap enough price and a big enough scale to where, we can make a lot of the things we want. But that always has not been the case. It's not always been the case, right? So there are these things that scientists discovered that are called restriction enzymes or restriction endonucleases. And what these enzymes do is they cut DNA at certain sequences, right? So if we go back to the DNA sequence and we look at NCO1, you see it's highlighted. It highlights the DNA and it says NCO1 enzyme will cut DNA at this specific sequence, CCA-TGG, right? So it's going to cut DNA at this sequence, and almost every time, it's going to cut DNA at this sequence. So if you purify and add this NCO1 enzyme to your DNA, it's going to cut it at that sequence, right? Nowadays, we don't purify these enzymes ourselves. Companies purify them and they sell them to you, and then, you just buy the enzyme from the company. But these enzymes allow you to cut DNA at specific places so that you can put DNA in, right? So like I said, if I want to put in a new gene, I'm going to cut before the gene starts, right? So we have our green fluorescent protein, which is coded in the DNA right here. Now, the NDE1 site is CAT-ATG. It's generally always used at the start of proteins because it has the ATG in it, right? And remember, ATG is the start codon. It codes for the methionine. CAT-ATG. And then you're going to cut something that's after the protein, right? Like perhaps this BLP1 or something right there. Now, when the sequence has the restriction enzyme site has an N in it, that just means any amino acid. Any nucleotide. So whenever you see an N in DNA, it just means any nucleotide, N, right? So this will cut at the sequence of GCT, any nucleotide, and then AGC. So that's the site, right? That's the site in DNA that this enzyme will cut at, and it will cut at generally no other sites. So once we cut the DNA out of this plasmid, what we can do is we can put in DNA that we have synthesized ourselves that will match up and fit right in like a puzzle piece, right? So now if we look at the Plasmid Map, what you can see is there are just a ton of restriction enzyme sites everywhere. These restriction enzyme sites give people the ability to kind of, pick and choose, to insert things, cut things out, do what they want with the plasmid. Now it's super primitive, right? I get it. What you're doing is you're literally changing the DNA just by cutting things out and putting it back in. But you have to understand also that plasmids are physical things, right? They are physical. So that means that you're going to have to interact with them differently than, say, like a digital system. So people love to use computer analogies to explain biological things, but it's, some of them are okay, but it's not quite true. It's not how it works, right? That's not how all this stuff works. It's physical. It's a physical thing, right? You can't just change a line in code and it'll change it. No, you literally, if you want to change the code, you have to remove something by cutting it out and add something by putting it physically in, right? But after doing that process, then you have this physical instruction set, which is DNA plasmid, that organisms can use to do cool and crazy things. All right. Any questions? We got any questions here? Anybody? Barry, you got any questions for me? What's going on? We need some questions. Questions, questions, questions. I know this is a lot of stuff, throwing a lot at you. But you know, try to just understand it as much as you can. So like, try to like take it in and understand as much of this stuff as you can. You're not gonna get it all immediately, and that's okay. You just need the repetition, the constant repetition to keep, forcing it down your throat and telling you how things should be. Now, the experiment that's going to take place is it's pretty simple. We already talked about most of it, but there's a couple things that I skipped. So let's go over that really quick. So the experiment you're going to do with genetic engineering involves adding, obviously DNA into the bacterial cell. Now, what you're going to do, the first step one, is to grow up fresh bacteria. Fresh DH5 alpha bacteria. So make some LB agar plates if you don't have any. Take your DH5 alpha stab and streak it out. If you don't know how to streak out bacteria, there should be a document about how to do that. You could also search it online. 2, what you're going to do is you're going to mix your DH5 alpha plus plasmid plus transformation mix. Right? Remember, so the transformation mix has the cations and it has the polyethylene glycol. So PEG plus calcium chloride, which helps the plasmid get into the DNA. Now, what you're going to do after this is you're going to try to introduce a rapid temperature change. Now, this scientists have shown helps the plasmid get into the bacterial cell or yeast cell. They're not quite certain why, but it actually works. So what you do is you put your DH5 alpha plus plasmid plus T mix into, say, your fridge for like 10 to 20 minutes. Not freeze or fridge. Freezing will kill the bacteria. And then what you do is you heat shock it, they call. You put it at a temperature close to 42 Celsius, and this creates a temperature gradient. Heat shocks the bacteria, and it helps the DNA get inside. Now, you don't want to go much higher than 42 Celsius because what happens is bacteria start dying as you start to go up in temperature. So you want to go 42 Celsius, and you want to go for like 20 to 30 seconds. If you go longer than 20 to 30 seconds, you can start to kill your bacteria. So be patient. Just sit there, count out to 20, and then take it out. It doesn't have to be exactly 42 Celsius either. Generally, what I like to say is if you have some like lukewarm water, if it's warm, when you stick your hand in it, but not so warm that it feels hot, it's probably somewhere around 40-ish Celsius. All you need is a little bit of heat shock to help this happen, right? Afterward, what you're going to do is you're going to help the bacteria recover, right? So we call it the outgrowth or the recovery period for the bacteria. And what you do is you add some media, so some nutrient-rich media like LB or SOC. In the kits we include LB, and you let the bacteria grow. So you want to put them at 37 C if possible, a comfortable temperature for bacteria where they like to replicate fast. Or if you want to leave them at room temperature, you just got to let them grow longer, right? It takes longer for bacteria to grow at room temperature, so let it grow longer. And then what you're going to do is you're going to do antibiotic selection, right? So after your bacteria have taken up the plasmid, they've started to replicate and recover, then you're going to put them on antibiotic selection plate. By this time, the bacteria that have the plasmid should have started to make the antibiotic resistance gene. So when you put them on the antibiotic selection, the ones that have the gene should live, and the ones that don't should die. So at the end, you should have bacteria with plasmid. It's not always the case. Sometimes you get contamination or something else, but that's generally what should happen. Bacteria with plasmid at the end. And that's basically the experiment. It's a similar experiment that you're going to do with yeast cells and human cells. The human cells, you don't do any sort of heat shock, but basically everything else is really similar. All right. So get on that experiment this week. Do your first genetic engineering experiment. Post pictures. Let us know how it's going. If you have any questions, as always, post it in the Google Classroom, and we'll do our best to answer those questions. Barry has a question. He said, how do you choose what promoters, origins, etc. to use? I know there are quite a few. Generally, there are standard plasmids that everybody uses. Generally, you just use those. Like, if you're starting to choose custom promoters, you're probably getting into really technical stuff. And so, once you get to that place where you're starting to choose promoters and origins, you'll have an idea of what you're trying to choose. Justin asks, if you don't do selection, will you just have a less pure result? No. So you have to imagine that only a small percentage of the bacteria are actually being engineered. So if you don't do selection, the majority of bacteria are not going to receive the plasmid. So your ability to find bacteria or whatever organisms that have been engineered is going to be almost impossible, right? Because in a small tube, there's going to be billions of bacteria. Billions. And you're going to end up with far, far less that actually get engineered. So you need a way to weed out all those billions that didn't get engineered. Otherwise, what you're just going to have is, right, low probability you'll ever find a bacteria that's been modified or engineered. You would never not use selection. Unless there are a few small cases, but let's just say you're generally never not going to use selection. All right, any other questions before we close it out for tonight? Anything else you want to ask about genetic engineering and plasmids? I'm here to listen. Try to answer your questions. I don't know everything. Alrighty, if there's no questions, then, I'll see you all next week. Make sure you ask questions in the Google Classroom or whatever if you have them. Otherwise, get those experiments done, and hope everything is going well.